My name is Sandra Kentish. I'm delighted to welcome you here to one of our seminars for the Victorian Division of the Academy of Technological Sciences and Engineering. I'm the chair of the Victorian Division. And I'd like to start our evening tonight by acknowledging traditional custodians on the lands on which we meet, which are the Wurundjeri people of the Kulin Nation, and recognise their continuing connection to land, waters and culture. And we pay our respects to their elders past and present. But we have a fascinating presentation tonight, the topic being microplastics and emerging contaminants. We have two speakers. They'll both speak for about 10 to 15 minutes, and then we'll have the opportunity for a discussion. So as they're speaking, think of questions that you might want to ask about this topic. So I'll introduce both our speakers first. So Dr. Minna Saaristo is an ecotoxicologist with over 19 years of international experience applying science-based approaches to assess the impact and risks of contaminants of concern on the environment. She's a principal scientist for land and contaminants at EPA Victoria and leader of the Emerging Contaminants program there. Before joining the EPA in 2019, Minna was a research fellow at Monash University for 10 years. And our second speaker is Professor Andy Ball, who's the director of the ARC Training Centre for the Transformation of Australia's Biosolids Resource, Australia's Biosolids Resource, and a distinguished professor. I nearly said you were a resourceful distinguished professor, but anyway, at RMIT University. He's also a program leader within the new Solving Plastic Waste CRC. And I forgot to mention this session was supposed to be chaired by Dr Ian Dagley, who is the new CEO of the Solving Plastic Waste CRC that's just been announced. Unfortunately, Ian couldn't make it tonight because he is away on CRC business in Brisbane. So that's why he got me instead. But certainly it is exciting to see that CRC become announced. And I think EPA is also involved in that CRC. So, sorry, to finish off your bio though, Andy was previously director of the Centre for Environmental Sustainability Remediation at RMIT and Flinders Bioremediation in Adelaide. So having screwed up totally all the introductions, I'll now hand over to Minna to give the first presentation.
Thank you, Sandra. Can you hear me all right at the back? Great. So yeah, I'm delighted to be here today to talk about the Emerging Contaminants program that we have at EPA Victoria. And I would also like to start by acknowledgement of the country. So I'd like to acknowledge Aboriginal people as the First Nations, First Peoples and traditional custodians of the land and water on which we live, work and depend, and I pay my respect to Aboriginal elders past and present. But I find that wherever I go, it's always good to start with definitions. So what do I mean when I talk about emerging contaminants? And they are, at EPA Victoria, they're actually a combination of new and old chemicals of concern. They are a variety of synthetic, but also naturally occurring chemicals in the environment. But what is common to these chemicals that they are the information about their concentration in the environment, their sources, their spatial distribution, and fate is very limited or sometimes even absent. But the key here is that the risk they pose to our health and environment is not yet fully understood. So you might have heard about PFAS. So PFAS, we categorize still under the emerging contaminants, even though we know they have emerged decades ago. And some of you in the audience might not know that we have such thing as Emerging Contaminants Program at EPA Victoria. And as Sandra mentioned, I started in September 2019, and that was when the program was launched. And the aim of the program is to monitor, identify, and assess the environmental and human health risk associated with these chemicals. But the key is really to guide duty holders to reduce the risk and prevent harmful pollution and waste. And if you are interested, we have a science roadmap that have been put on our website. So feel free to Google and see what are going to be the priorities for EPA Victoria moving forward to the next 5 years. But today I thought it's quite a tough topic to talk about emerging contaminants in 10 minutes. But I wanted to give you a bit of a flavor of the work we've done, and through those examples, walk you through what we have discovered and what might be the risk that we have identified, and also what are the next steps. So I take you back to 2019 when here was I would say the first study that we really, where we started with it was pretty ambitious. We went and sampled 245 sites in Victoria. So here you see a map, and every circle is a site that we sampled. And we sampled water and sediment, and also soil, and overall 101 water and sediment samples we collected, then 245 soil samples. And each of those samples we analyzed samples for PFAS, for pesticides, for phthalates, we saw plasticizers and metals. And if you're interested, there is a publication on our website, 1879, which summarizes all the findings. But the good news is that the levels were relatively low, and people always ask about the PFAS. So they were below the guideline values. But what we discovered was that these chemicals are in the environment, and they are related to a land use being urban and even some agricultural settings. So we needed to look further and where might be these chemicals coming from. So then we did a follow-up study where we were targeting biota, so in this case fish. And here again another map. So these are the waterways we targeted, and we were looking at what are the chemicals upstream from a wastewater treatment plant versus downstream from the wastewater treatment plant. And my background is in pharmaceuticals and endocrine disrupting chemicals, so I wanted to add those to the analytical suite, and then also look at, is there a bioaccumulation of these chemicals in fish? And the answer is yes. Again, nothing new. This has been found elsewhere in the world as well, but it's one of the first studies that really demonstrated this in Victoria. So showing just the pharmaceutical results. So out of the 72 pharmaceuticals that we sampled, 39 we detected in spot samples, and that means individual crab samples we took from the water, but also we used passive samplers, which we deployed to creeks and rivers for 28 days. So we had to take a spot sample on day 0, and another spot sample on day at 28 when we retrieve those passive samplers. And those passive samplers are those filters that will filter the water over 28 days. And being in the water for a longer period of time, they were able to detect even more pharmaceuticals. And out of the 135 fish that we analyzed, 16 of those fish had detectable levels of pharmaceuticals in their edible muscle tissue. And here is a diagram of, might be a bit small for it to see, but what it is showing that we detected venlafaxine it is an antidepressant. So antidepressant that is commonly used, but this is now one of the first studies showing that it is bioaccumulating in fish, and in this case in redfin. So if you look at the middle, I think I have a pointer. Okay. So if you look at this here, so at the discharge point, we had redfin that had up to 150 micrograms per kilogram of venlafaxine in the edible tissue. There's at the moment we did a human health risk assessment, and it's still you would have to eat a lot of redfin, that it would be a risk. So don't be afraid. If you go fishing, it's all fine, but it's definitely a concern. So therefore, seeing that kind of high levels in fish at the discharge point, and even at downstream from, and those two high levels were in the same waterway. So that definitely was a wake up call for us as well. We need to do further work to see why are there pharmaceuticals in the environment, and what could we potentially do about it. So then it followed with we went then to the wastewater treatment plants, and we started to sample incoming wastewater and compare that to the effluent, to the treated water. And we sampled 31 wastewater treatment plants across Victoria. And we again used spot sampling and passive samplers to collect the data and analyze the samples for pharmaceuticals, endocrine chemicals, and also of the industrial compounds and disinfection byproducts are used in it when you treat the water to level A, B, and C. And what we discovered was that, and this is again something that has been published, so if you Google emerging contaminants recycled water, you find that on our website. From 230 wastewater samples, a total of 413 contaminants were analyzed in each sample, and out of those 413, 180 contaminants were detected in the treated effluent, including pharmaceuticals, EDCs, and PFAS. Again, I know it sounds scary, 180. Again, the levels were below our current guidelines, but it's still an indication that there is a mixture of chemicals present in our treated effluent. And the good news is that for most of these chemicals, the levels were lower in the treated than in the incoming untreated. And the highest concentrations were substantially decreased, but there were some compounds that were passing through the treatment train untreated. And one of those were some of the pharmaceuticals we found in the fish. So now we are going back to looking at the chain, that where are these coming from? So clearly wastewater is the wastewater treatment plants, not all of them are capable of or equipped to treat these new emerging contaminants. But again, good news that we were comparing different treatment technologies that these treatment wastewater treatment plants are using, and there is one of them is, we call it the treatment train 12, which is using a combination of activated sludge process with extended aeration, UV, light disinfection, microfiltration, reversible osmosis, and chlorine, and it's a gold standard. It's a very expensive, and it's not going to be feasible for setting up for every single wastewater treatment plant in Victoria. However, it's good to know that we do have technology available that in certain circumstances maybe is something that we should consider as especially in high risk areas. But again, people ask about the PFAS, the concentration of PFAS we detected in the effluent way below the human health guidelines. So yes, we are finding these, but at the moment the level seems to be low enough that we just need to be monitoring and seeing if they change over time. And moving on then, yeah, we're done baselining in the sediment, soil, and water. We looked at what's in the fish. We went and looked at the influent and effluent, and then we got opportunities to look back in October, December 2022. You remember the big floods that we had. We had an opportunity to look at what happens when big areas are flooded, with the chemicals travel with the flood waters. So we had a collaboration with the Natural Hazards Research Institute and Victorian SES, and we the aim of the study was to identify hazards and contaminant risks associated with flooding and provide evidence-based advice to support community actions. So there's a map again, all the sites that we sampled, and some of them, the 19, 20, and 21, where actually we needed to use helicopters to transport the sample bottles because all the roads were closed. We couldn't even get there. So thanks to our collaborators, we were even able to do this kind of sampling campaign during the floods, and we revisited 21 sites 3 times. So it was a really incredible effort from all the partners. And what we found, surprise for us, yes, we found contaminants again. But of the 780 contaminants that we analyzed in each sample, 81 we detected. We found fungicides, we found PFAS, we found phthalates, and we found polycyclic aromatic hydrocarbons. But again, levels were below the current guidelines. So yeah, low levels, but clearly they are moving. And one interesting what we found was phthalates, which are used in plasticizers, because some of these areas that we sampled are really remote. They are in a regional Victoria. So it was quite interesting to find really chemicals that we normally find in an urban environment, even in those flood waters. So it was a reminder for everyone that, we always advise is avoid going, the flood waters are dangerous. Like, please don't enter, even though we always, first we think about is pathogens and bacteria. And we did find that there is actually the image here shows that the E. coli exposure is the post-flood is actually the point when the levels are the highest, but also the chemicals are traveling there as well. So it's best not to enter, don't go canoeing or swimming. So definitely we got a lot of questions like, is it safe yet? Like, no, actually it's not safe yet. So it does the flood waters to move chemicals around. And the last example I want to show is the work that we are currently doing, which I thought would be interesting. So we're looking at what about the lettuce and broccoli that we eat every day? Would we find any chemicals in those? So the aim of the project is to determine the concentration of chemicals in irrigation water and soil, and also in broccoli and pasture, and investigate the potential for these contaminants to bioaccumulate in plants. And we have sampled 6 farms in Victoria. We chose that we have 2 farms that are using broccoli, and then we have a reference farm that doesn't use recycled water. We know the reference, it's challenging. There's no such thing as clean site. There's always some background, but what we wanted that none of these sites have used biosolids, so we at least knew that there shouldn't be that kind of chemical residues present. But what the difference between this treatment and untreated was that the reference sites did not use the recycled water. And 173 samples were collected, 50 water, 36 soil, 87 crops. We sampled different parts of the crop, meaning the roots and the leaves and the florets, and we analyzed them for more than 1,000 chemicals. We collaborated with the CSIRO in Adelaide, and they detected 65 chemicals in water, 160 in soil, and 100 in plants. Again, the usual pesticides, some pharmaceuticals, and phthalates and PFAS. And this is results that I just got a month ago. So therefore, I'm pretty giving it at a high level because we're still analyzing it. But what is becoming obvious that there's actually no big difference. That the difference between using recycled water and the water from the river is very similar, which indicates, as the studies that we've done so far shows, that it might be actually the chemicals might be coming from the river water more than from the recycled water. So we're going to do a follow-up sampling. We're going to go back to those farms now in April, May, and do more sampling around the areas, like what are the other potential sources for these chemicals, because it's not really clear to us yet where the chemicals might be coming from. But the good news, again, all the levels are relatively low. Please do eat your lettuce properly. I think it overwins the risks, but definitely chemicals are there, but we are working on just finding out where they're coming from, and could there be some ways that the farmers could maybe time the use of the pesticides in a more targeted manner. So just to finish with some maybe some key messages and things that we're discovering. So yes, as you're hearing, the emerging contaminants are frequently detected in the environment, but at the low concentrations. The challenge that we're facing is that we have really, the risks are still relatively unknown for many of these chemicals, and therefore we don't really have a lot of guidelines for them. And more finding that traditional endpoints, the way we assess risks, looking at growth and mortality, might not be the best when we talk about pharmaceuticals, which are known to affect behavior and other population level effects at very low concentrations. And the key is to have long-term monitoring programs which will be able to detect changes over a long period of time. For example, when the flood study was done, it would have been ideal to be able to know what were the concentrations before the flooding. So we could know have the conditions improved or become worse. And lastly, one of the future directions, for at least for EPA Victoria, is collaboration. We need partners. We need to work together on this. That it's such a challenge. There are thousands and thousands of chemicals, and even though we are doing our part, but we need to work together to provide better solutions and better manage and then translate that science into policy and decision-making tools. And we are working with other government organizations to develop a framework for emerging contaminants, but it's still in an action plan level. So hopefully the funding will come and we have a nationwide framework for that. And also I would, I don't see a lot of students in the audience, but it's very important to remember that we need scientists, but we also need people who translate that science into policy. And often I find when I work at the policy part of our organization that the language is not always there. We scientists, we always think that the data is enough, but it's not often enough to have the data, because it needs to be translated in the right way for the policymakers to really, I would say, get what's the risk and why we need to do something about it. So it is a whole nother, I would say, definitely any professors here, I really highly encourage students to be considering that. How do you communicate and how you engage the non-scientists? Because I find that there's often the gap is still too wide. And finally, it's important to remember that everyone can do their part, especially when it comes to pharmaceuticals. What you do with your expired medication really matters. If you just flush them down the toilet, they will end up to the wastewater treatment plant. They might not actually treat them at all. So therefore, please do, don't forget that everyone can do their part, and together we can manage the environment better. So thank you. That's the end of my presentation.
Thank you, Minna. And we will not take questions now. We'll let Andy talk and then we'll have a broader discussion. So over to you, Andy. Yeah.
Okay, good evening. Thank you for the invitation to come and speak. And thank you for coming along and listening to Minna. Fantastic talk. And I'm going to talk a little bit about microplastics. And in particular, just going to introduce the what, where, how, what's the risk, what do we need to do to mitigate the risk. To give a really brief introduction into the world of microplastics. especially those that we find in the environment. So first of all, what are they? Well, they're really plastics that are smaller than 5 millimeters, but larger than 1 micrometer. After that, we move into nanoplastics in diameter. They're not always round, but some of them are, but as long as they fit that size, they're microplastics. And there's 2 sources of microplastics, primary and secondary. Primary are designed to be small. They're there for commercial use. They're used in cosmetics or they wear. They're the microfibers that are shed from clothing and textiles. So they're the primary microplastics, but secondary microplastics, just as important. And they started off much larger particles, but as a result of breakdown of the larger plastics, water bottles, plastic mulch films, pretty much anything that's out there in the environment, they will be broken down to smaller and smaller fragments, not degraded, just broken down, and they will form microparticles, microplastics. So the environmental factors, obviously solar radiation, any physical process, so that might be a wave action if the plastic's in the water. It might be ploughing if it's in a farmer's field or in a horticultural scenario. So there's primary and then there's secondary sources. How much is there out there? This is a really topic that's always changing. In fact, these are fairly conservative numbers, but there are in the environment somewhere between 12.5 and 125 trillion particles. Others will say it's much larger than that, up to 10 times larger, but I think that's a pretty conservative number. And they're in every compartment of the environment. They're in water, they're in the air, they're in the terrestrial system. So in the ocean, so there's between 15 and 51 trillion pieces of plastics. And that weighs up, ending up to 236,000 metric tons in our oceans. Air is not immune either. So some workers at Utah State University collected samples of air from 11 and rainwater from 11 different national parks and wilderness areas within the US, the Western US actually. And they found enormous amounts of plastics in the rainwater. And in fact, at those 11 sites, 1,000 tons of plastic was recorded as being transmitted through the air. Soil, really hard, really an unknown factor in many ways, especially in Australia. But 13,000 plastic items per kilo of dry soil, which is about 4.5 milligrams per kilo of soil. So plastics are truly everywhere. They're already there. In Europe and North America, it's thought that agricultural systems probably have far higher quantities of microplastic than oceans, up to 3-fold more in the soil than in the ocean. Although oceans rightly had a lot of focus, it's much harder to detect microplastics in soils, as you'll see little flecks of microplastics, but it's pretty hard to actually start to account and identify them. So we have a lot of work to do. So can they transfer through the food chain, these microplastics? Yeah, there's more and more evidence now that both food and water is contaminated with microplastics. This little figure here shows a result of a study in Italy when they're looking at microplastics particles in various vegetables and found them in all of them, all fruit and vegetables. Research has also shown that wheat and lettuce, when planted in soils with plastics, actually can take up those particles, albeit a laboratory study at the moment. But they showed that plastics could be taken up, microplastics. Scary. And obviously the main source of microplastics in our fruit and vegetables is likely to be the soil from the uptake. microplastics are not just in the vegetables and the fruit, they're also in the things we, in higher animals. So microplastics being found in blood samples. In fact, every sample that the Dutch group took of pigs and cows' blood identified microplastics. And of course, we have indeed identified plastics in human blood too. And of course, in our aquatic environments, many studies have shown microplastics in fish muscle, not just the gut. So it's certainly things that we are being exposed to. So microplastics are there, they're out there, and we can't avoid them. We're in contact with them constantly. So it's not surprising that microplastics get into our bodies. And in fact, there are two main ways. One, as you might imagine, ingestion, eating some of this contaminated fruit or drinking the water, but also, of course, inhalation, because the microparticles are in the air. So microplastics have been in the human lung, maternal and fetal placental tissues, human breast milk, and of course, blood. We will come on to a caveat here. But what we don't know is how these particles are processed by the body. How often do we shed them? Where do they accumulate? We need a lot more work going into that exposure. There's a nice study from using tissue cells, human tissue cells, exposed to nylon fibers. And these tissue cells went on to form lung tissues. And there was a reduction in the number that formed and also the size of the airways. So it does seem as an interaction. But the interaction may not just be because of the plastics. I think, as Minna suggested, there are other components in the plastics. So things that are used to harden a plastic is an endocrine disruptor. Phthalates makes plastic soft and flexible, and that affects fetal development and reproductive problems in adults. And obviously plastics, and some of them are derived from electronics. And again, they are have impacts on human system. So there's a lot of microfibers, and generally microfibers are the most important components, rather than the smaller particles. Okay, so what are the gaps and who's doing something about it? Well, in Australia, there's a lot of research going on by a number of bodies, and I've highlighted two, of course. The CSIRO have been running a great program, and about that's the wrong way around. That's thrown. That's their website, and of course the Solving Plastic Waste CRC that Sandra was talking about just being funded. And indeed, we have the chair with us, Leonie Walsh. We also have Matthew, who is heavily involved in licking us into shape, ready for the interviews for that CRC. So both will know a lot about this. And although the CRC has is looking at both producing new types of plastics, bioplastics, recycling more effectively the current plastics, looking at the social aspects, life cycle analysis, the area that I'm involved in, these are our goals, and I think they're pretty much standard amongst anybody working in microplastics. One of the biggest problems, you saw all of the numbers, but there is no standard protocol. There's no standard way to collect, to isolate, to quantify and characterize. Everybody is doing different approaches. And there's no international standard, there's no national standard. So that's something we really were going to strive for, working internationally to try and get standard protocols. Now, there is lots of different ways of doing it, using Raman microscopy, the pyrolysis, GCMS, but none is perfect. It depends what you want. But nevertheless, we have to arrive at a number and a way that's standardized, because I'm aware of research looking at a number of microplastics in drinking water bottles. And I know of one study that reported 248,000 particles per bottle. And I know another group who followed up that protocol and got 100 times less with exactly the same bottles of water. So there is a huge diversity in these numbers. And I think we need to strive. As you can imagine, microplastics are everywhere. So you really have to have very stringent conditions like PFAS to measure these things. And we're not doing that very well. And a lot of publications are being questioned in terms of what were the controls? Were they sufficient? So we do really need to do that. And that's one of the first things we'd like to do in the CRC, because without that, it's very hard to really understand the true risks. But then we need to source, fate, and management of microplastics. So for the CRC that we have, that will involve actually something our citizen science project. So we will have citizen science and rangers in indigenous areas will be taking samples, farmers, rural areas to look. We have more microplastics in suburban soils and in cities than we do rurally, but where you have a lot of horticulture, you use a lot of plastic in agriculture, you tend to see hotspots. So we really do need to understand what is in Australia, what is the number of plastics we find and where are the hotspots, and therefore where can we focus most attention to try and alleviate this issue. And then truly the risk of microplastic pollution on humans and ecosystems. If you're looking at risk, you really do need to understand this, especially for my interest in ecosystems. How does the plastic move between, for example, the soil and the groundwater, or from a trophic structure? And then lastly, a whole new area of, well, we've got great plans in these projects to replace plastics with much better plastics, with biodegradable plastics, etc. But we have to deal with what we have now. And so we have to put mitigation strategies in place that are effective. And so there's a whole area that we need to do on can we develop the enzyme technology? We know we have PETases that are starting to be more and more effective with genetic manipulation. Can we try microfiltration? Can we try some other electrochemistry? So we need to develop solutions that where we do have hotspots, we can deal with them. I think that's analogous to those contaminants that Minna was talking about. So that's where we are. We're really at the beginning of a journey, and certainly those of us in the CRC feel that we've got a 10-year program to try and make some inroads into this process. Thank you very much.
Now we might get our two speakers to come and sit over here. But thank you both. I think you've scared a lot of us more than anything else. You can sort out the microphones. I'll stand here. Can you? Yeah. Yeah. So we are opening the floor for questions, but I'm going to probably kick off first. I'll turn the microphones on. I suppose I've got a question for both of you, which is as we move towards the circular economy, we're supposed to be starting to recycle more and more. How do we prevent the build-up? I mean, it's quite frightening to think of how much is already there. How are there strategies to prevent the continuing build-up of these contaminants in the environment? Is that possible? So I'll give you guys Minna a microphone first. Hopefully that's on.
I hope so. Yes. Can you hear me? Yeah, good. Yeah, that's a big question. And first thing, I'm a toxicologist, so I am very mindful of the risks of recycling. But equally, I know that we need to, we cannot just be creating new products, so we need to recycle. So talking from the EPA perspective, it's all about this understanding the risks and trying to work towards and think about the life cycle, like what happens to the product and how, and what is, and what are, go to the basics, what are the half-lives of these chemicals and how do they respond to different conditions? And it comes back to the engineering and thinking about the life cycle of these chemicals that are in these products from the beginning all the way to the end product, which actually, I don't know, do we always know the end product? Maybe we're not always mindful of where does it end up as with the plastics. So, and I know the examples with the tire rubber, that we want to be recycling tire rubber, but now there are studies showing that there are chemicals leaching from the tire rubber, which is affecting salmon and organisms that are living close to roads where we are adding. So they are, I'm all for the recycling, but I think we need to be mindful of the potential risks of maybe using those materials in the conditions that they were originally designed for.
I echo his thoughts there, and one of the interesting things of that that's just emerging is that the idea of recycling plastic is obviously not new, but you know, if you haven't got a well thought out chain for the processing, then it becomes a problem, as we've seen with Red Cycle. But you know, there are proposed to be some new large recycling facilities in Melbourne, and one of the recent research has shown that any recycling of plastics generates huge amounts of microplastics. And so we do have to be careful and understand, again, the risk and mitigate that risk, even when we're trying conventional systems. So, you know, recycling is good, and developing, you know, biological, biodegradable plastics might be useful in certain circumstances. You know, I know that a lot of people are looking at developing plastics that you can break back down into their monomeric units and then reuse them and produce something equally as strong and effective. So it's not a downward spiral, which is a good thing. So I think there are numerous strategies, but we are left with microplastics in the environment now. So we're reaching a system where sustainability and sustainable, you know, circular economies are might be compromised. So, for example, the work we do with biosolids and composts, these are things which are really proactive. Yet the idea of having contaminants in those products is beginning to lead to questions about whether they are sustainable. And we really don't want to get to that stage where you can't put them where they, you know, they're doing good. So we do have to mitigate trying to wrap the plastics as soon as we can.
And Leonie, so it looks like she's got her hand up there.
I was just going to add to that too. At a federal government level, there is a working group looking at national packaging design standards across all of the packaging materials, and they're looking at what are the right levels of recycling that they're going to mandate. But they're also looking at labeling, adhesives, fillers, and putting standards in that take all of those into account. So, and that the work's being done across the EPAs, across the states, across the packaging councils, the retail groups. So I think it's the first time that they're trying to get harmonization and consistency and have something that's also traceable for imported materials.
Yes, I could keep going myself. I'm going to ask Sally's got a quick note. I'll get Matthew first. Yeah.
The question's for Minna, really. I'm very intrigued about the antidepressants in the fish. And when you said that, why did you look for it? So I guess the question is, how do you decide with all these hundreds of things you test for? And where's all the analytical capability to test for those things?
Thank you. Yeah, of course, I had only 12 minutes, so it was a very brief overview. Why pharmaceuticals? So as I mentioned, my background, I've done decade of research on looking at how pharmaceuticals affect fish behavior. So maybe I could use the word bias. I was biased based on my research background because I knew that nanograms per liter concentrations will have adverse effects on reproductive behavior of fish that I've had in the lab for that long. So when I was given the opportunity to do a field study and explore other emerging contaminants, I proposed that how about we look into pharmaceuticals as well, because I know based on my research, which is published in high impact journals, that they will have effect and is likely, and often I get questions about, is it environmentally relevant? And I couldn't reference a study. I couldn't reference a study that would show that these concentrations in Victoria will have effect. So then when I went into the field, yes, it's not as simple as I want to study this, who can measure it? So I did do market research on what lab could I use and what lab could go to that concentrations and that low. And as you saw the numbers, we analyzed more than 100 fish and only 16 had detectable levels of pharmaceuticals. And for me, that shows, I bet there was a lot more, but the limit of reporting was not maybe low enough. So my honest personal opinion is that I think we actually would have had even more individuals with pharmaceuticals if the method would have been. So method was able to detect these micrograms per kilogram concentrations, but not. So, and that work was done 3 years ago. So I bet if I would go to the same lab now and support commercial supply, they would be able to, the methods are being developed all the time, which is good, but measuring chemicals in tissue samples is not a small task. It's a very, and you need to have really, again, and that's why when we had the 150 micrograms, I didn't believe that. I called my colleagues in Sweden and said, what's going on with this creek? Like, am I dreaming? And then they were also like, has someone been just like, pouring? So it was a very high detection. And in the end, it was the highest redfin we caught. And as I said, we went back to the lab and I went through the Q and QC, and it was, I trusted the result. And then it made more sense when I started looking into that there's actually other fish in that riverway. So it might have been just a one-off. It might have been, if we go back, I don't know, would we find that again? But it was definitely a single that maybe something is in that area that is causing that. And redfins do travel. So I don't know what they might have been able to accumulate from others, but we didn't really see it upstream. So therefore it was pointing that it is something that's coming through the wastewater. Answer there was like, why pharmaceuticals, who measured? But yeah, it's always a journey. But maybe it's overall, why do we measure what we measure? It comes down to a budget, how much money we have available, what can the commercial labs, what can they detect as well? Like what if we have big, so when I say PFAS, at the moment we measure like around 70 different PFAS compounds. When it comes to pharmaceuticals, when we do targeted analysis, it's like a suite. It will be, it's not one, it's whatever they have in their standard suite. We are moving towards non-target. My aim is to, because there's so many that we're not even measuring. So I would like to look for fingerprints and to look for signals. And then once we see that there seems to be class of this type of compounds, then go to the targeted. So we are definitely moving towards that, because if we always go look at pesticides and PFAS and EDCs and pharmaceuticals, we're likely to measure, like miss all the others that are out there. So it is an ongoing process, and we always, work closely with colleagues that I know from my research years that we know that, and CSIRO has been fantastic with that uptake to crop study, because they're going well beyond the original project scope, because they're interested, they want to know as well, and they're developing the method while they're analyzing our samples. So hopefully next time you hear me, I will maybe have even scarier results, like, hey, we've actually 5,000 chemicals now.
I think Sally's got a question for you.
Please no. Terrifying enough. And actually, coming back to this point of when you start to look at these microplastics, Andy, is there a situation where even the biodegradable plastics will form microplastics simply because there's less plastic and it's all the other stuff it picks up? So I think of it as a colloids and surface scientist, that's a lot of surface area. And that could stabilize itself. And so it doesn't matter if it was biodegradable or not, it's just small enough to be coated and now not degradable.
Yes, I mean, I think, true biodegradability is probably, it's not an issue, but what I didn't highlight is that, the nature of plastic, you're absolutely right, means that other contaminants readily absorb to plastic. So you get a lot of those contaminants that Minna was talking about stuck onto your plastics, creating even more of a problem.
Minna and Andy, thank you very much for the talk. And I can tell just by listening to the presentations, there's still a lot of understanding the size of the problem here a little bit. And you talked about like understanding the trends and further work needed. And Andy, obviously the CRC and CSIRO are trying to get a handle on just how significant this is. So can you, in that context, as a regulatory scientist and an academic, can you give us some comments about like what you're seeing of industry's role in understanding the problem, and maybe even further commentary about what your thoughts are, what industry should be doing to help with not only understanding but mitigating coming forward?
Start, yeah. Good question, and actually, I'll be grateful that through my work now at EPA, so the effluent study that I showed, it's a collaboration between water industry and government, so we already working together, because as I said, industry, you know, and of course, regulator being a part of course, they want to help us to, you know, at least go to the root of the issue so that we can work together. But I would say we all have responsibility, and now under the new act, and we have the general environmental duty, all the Victorians, every single one of us actually has a role to play here. And that has become very clear to the industry that they cannot no longer just look the other way, that they need to be mindful and they need to assess their risks. And that is not a simple task. How do you assess risks of something you don't really understand? And there's a lot of uncertainty. So we are, and I have to be, this is my personal view with my colleagues here, we're all learning and we're all working together. We talk about we are on a journey, but we are literally on a journey of like trying to work together towards having a better understanding of the risks associated with chemicals, because I would say, even in this 5 years I've been in my role, we know a lot more about PFAS now than we did 5 years ago. We are able to detect, we're able to have a better understanding of the scale of the issue, but it's, yeah, small steps, but without the industry, and industries, I really, you know, it's very important that we listen to industry as well, because sometimes as a regulator, we might be proposing something that is actually not, like there's a management strategies, or how do they irrigate, or how do farmers even, when do they use the recycled water, really plays a part. Like, let's say that there is, and do they use the recycled water, or do they use the river water? So it's very important to listen to both sides and try to together try to get to the root of the issue. And also, like, what kind of products do we design in the first place? So it is complex, but I think the industry really is like, I think, part of the puzzle.
Yeah, great question. In terms of microplastics, I think the industry is torn because they have, you know, obviously they're aware of microplastics and they understand that it's a potential problem, but it's really expensive to analyze. It's, you know, $2,000 or $3,000 a sample, and the message we get strongly is really interested. We want to do something about it, but it's not legislated. We don't have a limit of plastic particles in our drinking water or wastewater. And therefore, you know, the most urgent thing are some of those other contaminants. And that just has to sit in the back of the queue until there comes a time when somebody's going to say, okay, there's a legal limit. And in other places in the world, in the US, there is now a limit on microplastics in drinking water. But that obviously precipitates more interest. So at the moment, there's a watching brief on many industries who realize that they have to do something, but there's a queue of other contaminants which are being legislated against, and they need to put most of their efforts into that, PFAS being an obvious at the moment. So I think there is industry is definitely torn between, you know, other microplastics, for example, and other contaminants. Thank you. Other questions?
Oh, sorry.
Just following on from Michael's question about industry, I just wanted to talk about the regulators, but not necessarily down at EPA level, but in terms of chemical safety testing. So most chemicals and chemicals have got requirements for environmental fate testing. So do you have any sort of feeling that an environmental fate test could be refined more for us to predict pathogen further in the environment? And one for Andy is, how reactive is this microplastic? Because that's really what's going to drive a regulatory reaction to it. I remember 1,000 years ago having to look in a food application for edible plastic that was being put onto reconstituted orange juice to make it look like we picked the oranges, and it was inert. But the question is, morally, you know, what is that doing to misleading a consumer? So, you know, way back then, plastics were being introduced as being non-reactive, but where do we think we are now? Sorry for the true from, but you can resist.
Thank you, maybe I probably will share my personal view on this, because I'm part of the EPA science part of the, so I'm a scientist in the regulatory space, but yes, I'm aware that I'm part of the National Chemicals Working Group where I work with my colleagues in different states and territories, and we do, discuss limitations and improvements, and there is initiatives at the federal level and ICAM and definitely their regulations. But I would say at the space where I work is the emerging contaminants, and some of them are because of the improvements in technology, might be the fate and transport at the level that when we thought maybe a decade ago was safe, but now we have a better understanding of the technological improvement, but actually we thought that they would not be traveling they wouldn't be so mobile, for example, or they would be more, they would break down, but now we're finding that they do, and they actually create metabolites that are even more. So I think the chemical landscape is still a bit more complex in certain instances, so therefore I think they are, and my colleagues are full aware that we need to stay on top of what are the research, what's the latest research showing, so we can then keep on reassessing and be more agile, because if we just trust what we thought that was the best science 10 years ago, it's likely to be different today. So it's a work in progress, but yes, it's a challenge, but my colleagues are well aware and we're trying to do it together. But I think key is the information sharing and knowledge sharing as well, so we don't try to all this work in silos, because it's such a complex landscape.
Yeah, for me, in terms of how reactive are microplastics, I think the chemicals associated with the microplastics are probably the most reactive component. But there's enough, I think, evidence out there to say we should take a precaution principle. We need to do more work on that. That's understood, but there's enough insights to learn that it is definitely something we need to focus on. And for example, there's a recent study just a few weeks ago that identified that plastics, just plastic particles, were at the basis of a number of clots in blood of, in a study of rats and mice. So again, I think even its reactivity may not be so important as the physical particle that may be in the bloodstream.
Michael.
And so, I'm working in water treatment and water recycling area. I'm kind of used to scary stories. So, so one that kind of alarms me a bit in, seeing what's happening in, using circular economy, as Sandra mentioned, for reusing the water and closing the loop, is antibiotic resistance. So I just wondered if you can comment. I don't know, microplastics, there could be concerns with that, but definitely in the trace organic compounds, like in pharmaceuticals, you're, maybe do you include that, or do you just sort of lump them together? But that to me seems like one of the more tangible concerns. I mean, PFAS, we just don't know. I mean, they are scary, and maybe antidepressants and things, but small quantities. But the antibiotic resistance and threat that we suddenly couldn't use antibiotics, for example, we can go back to tuberculosis days. So I'm just wondering what the EPA, or even if you could tell me, there could be a scary story in microplastics as antibiotic resistance or antimicrobial resistance. So that, just want to comment on that.
Good question, and thank you. It was actually, the good news is, yes, we're looking into that as well. It was part of the uptake to crops study. So CSIRO has they work on AMR and they had research in Brisbane. So we have, I didn't include that sentence, but we found AMR. So we found inedible parts of, again, the reason maybe why I didn't want to bring you to this, that we're still working on what do the levels that detected mean in terms of safety, because it is still a new and evolving. But yes, AMR is definitely something that we continue looking into. It is a little bit more challenging from the analytical point of view, because there are a limited number of labs that can do that. But through CSIRO, we are definitely going to be doing more and more, because it links to the chemical structure as well. So we know that certain pharmaceuticals in particular and certain chemical mixture will actually, it's likely to increase that. So it's unfortunately that those two are interlinked. So there will be if we ignore the the existence of AMR, we are unlikely to be solving it, but it is something that it's only, I would say at least at the regulatory level, I believe we are the UK is one of the first that have started to include that to the analytical for the monitoring programs, because we're aware of that, but it's a bit like, how do we measure it? Who can measure it? It's not a kit that we can buy, so it's still, but definitely we are working, we're part of One Health consortiums and funding, so we definitely are, we're working because, as you said, it is such a enormous and scary, like what might happen if we just not look into that, but I believe there should be a program coming next year that in Victoria, but it's something that we've kind of done as a nice important thing to add on.
Yeah.
But it's a little bit on that more in the discovery space still.
Yeah, it's in discovery space, and even like, a lot of water treatment processes won't actually, like, for example, if you're looking for a particular antibiotic, for example, water treatment processes are really good at smashing up molecules, but those functional groups can still persist. And so sometimes the molecule might not be there, but its effect can still remain. So a little bit concerning. So yeah, but really good job looking, getting on for it.
And Andy, do you want to add anything?
Yeah.
Well, to the best of my knowledge, I don't know any work that's been looking at AMR genes and association with microplastics. But I guess wearing my Transforming Biosolids Center hat, then now I'm really interested to see that we just completed an experiment where we've been looking at the impacts of adding various amendments to soils in terms of AMR genes present. So we've just done a metagenomic analysis and just starting to analyze that data using the biosolids, using compost, using biochar, biosolids and a control. So looking at the number of AMR genes that are in the soil, even in the plant rhizosphere. So that'll be exciting when we see the data.
Now I'm going to ask a question, but then we come after that for one more. So if you are thinking about having a question, put your hand up or something. But my question is, how do we compare to the rest of the world? And I mean that in two. Again, I'm going to have double question. How do we compare in terms of our ability to do the science, so to detect these chemicals and to perhaps understand their impact? But similarly, are our rivers and waters just as polluted as the rest of the world, or how do we compare in terms of our level of contamination? It's horrifying to hear that remote rivers have got phthalates in them. Is that common for the rest of the world? So yeah, just generally the global view. I don't mind to go first here, and I'm holding out the microphone for everybody else. Keep going.
So it's in terms of the pharmaceutical, I would say we are as polluted as, we're not worse, but there are certain things that we're picking up, such as there are certain antibiotics that we use. Actually, antibiotic was one that cephalexin that was coming up in our monitoring, where the levels were at the microgram per liter level, and that was a bit higher. So there are certain ones that are just, maybe we're using them differently or more. So there are certain kind of differences, as is always is the case. But overall, what we're finding is similar to fingerprint chemicals is similar. So we are, definitely, unfortunately not better, but I would say not worse. When it comes to ability, I haven't worked in any, to be honest, like as a regulator, I feel like I do have enough, but I think I am lucky that I know almost everyone who works in this space, so I know where to go when it comes to Australian, if I need someone to work on pharmaceuticals or AMR or PFAS, but I would say there's still room for improvement. So I would say competition wouldn't hurt. It would be good to have more commercial labs, because the analytical costs are still pretty high. So that's why I would say there's space there. So hopefully then through that turning on to duty and the industry coming along, there will be more push for, because I would like to start adding bioassays and looking at other ways of looking at more the effects of these chemicals as well. So in that space, when it comes to bioassays, we're quite limited of who can supply. If I have 200 samples, would I send them to not that many labs? So again, a bit of a yes and no answer to that question.
In terms of microplastics, I think the technology is here in Australia, it's equivalent to anywhere in the world. In terms of how our soils and waterways are, certainly we're probably not at the levels generally that Asia or North America or Europe are at. But then we have, specific areas where, for example, horticulture uses huge amounts of plastics. And traditionally they're left in the soil and they start to fragment to become microplastics. And horticulture innovation and partner in the CFC, and they're very aware of that. So trying to find alternatives to that. So I think there are going to be hotspots where we will find significant amount of plastics. But generally the soils and waterways are not as polluted in terms of microplastics as Asia.
I'm glad we're going to be able to end on a somewhat positive no, Robin, don't spoil it.
But thanks to all, and if I could just build slightly on Sandra's question, actually, toxicology. One way or another, you've both mentioned that there's some big gaps here in our knowledge. How do we get more international work going on the toxicology side? Is there a role for the academies, for example?
Wow, yes, as I said, my background, I did a lot of mesocosm and toxicological studies back at Monash, but comes back to funding. So how do we, I would say government can be a driver as well for that, and that's what when I visit the framework that I am working with my colleagues towards building, working to get together that we because again, some of these analytical costs for analyzing 1,000 chemicals in a sample can be $3,000. So if we can, through research or through partnerships and through consortiums, or so I would say work together to try to funnel the effort and then and the funding required to create that, I think that will be beneficial. And that comes to toxicology, doing that, looking at the adverse effects, and because we know that some of these we don't know the effects, so it takes a while. It's not going to be a 24-hour quick essay that will provide you the solution of multi-generational effects and adverse effects. So it's going to be complex, but at the same time, I'd rather see us putting the effort in one basket than trying to all do in isolation and then publishing studies that unfortunately often the regulators don't even find, and we don't know what's there. So I think collaboration is key, and then definitely big funding bodies as well to work with this.
I can just echo sentiments, and toxicology is interesting because there are standard protocols, so we can apply international standards and do those things. So I think it has an important role to play whilst we're developing standards for microplastic detection, for example. So yes, well, good point.
And I think we've run out of time. So if we can both thank both of our speakers again for really interesting presentation. Although somewhat a bit depressing. And profile our event next month. And Sally's looking at me like she wants to advertise it.
No, I'm just saying. Just profile it.
Well, I'm trying. Is the topic specific? You
Yes.
can talk the topic because I'm not sure I've got the exact topic. I know we've got one of our speakers here.
Excellent. So our topic for May is on AI for digital health, but also for social good. So bring all of that together to get beyond just the digital health story, to get to equity, equality, and all of the things that are coming around that we can use AI to actually help us with. We've got 3 brilliant contributors to it, including our moderator from Melbourne University, and with Wendy, and then Karen Verspor and Joanna Batstone from Monash. So
He would
great
snap.
lineup.
Yep. Thank you, and we will end there. For those of us having dinner, please have some dinner. Thank you. Thank you.